what I'm going to talk about is some background, biologic background to the basics of understanding bone formation, bone regeneration, and talk a little bit about the biological challenge of guided bone regeneration. And I hope actually it fits in quite well with the previous two talks, which I enjoyed enormously, and I thought would sort of rather set me up quite nicely, because I think everything that was said and all the different methods that talked about fit into the biological model that I'm going to talk about a little bit. And probably the most important reason I've been asked to come is that our lab has been doing some work in collaboration with the Translational Cell Biology group at Geistlich. And we've been doing some work on the interaction between cells and membranes. And some of that data turns out to be quite interesting, even if we don't know its clinical significance. So I hope you'll find this is a short diversion from the largely clinical stuff. So firstly, I want to talk a bit about bone biology, back to basics. And if we start off with the cell that makes the bone, it's an osteoblast, you know that. And the thing that an osteoblast does is it secretes bone matrix proteins. And that's basically how it makes bone. The important things about osteoblasts, like any other cell, particularly when you're thinking about wound healing and tissue regeneration, is that these cells tend to be what we call post-mitotic. It means that they don't tend to divide, and they have a finite lifespan, which has been measured in rodents of about 28 days. In humans, it's estimated to be about 3 months. So there is constantly a need for replacing these mature cells because they die off after a certain amount of time, or they get incorporated into the bone matrix to become osteocytes. And of course, these cells come from stem cells, so-called mesenchymal stem cells, during normal turnover, and particularly in wound healing, where there's a sudden demand on big influx of cells to make new bone. And we talk about stem cells a lot. Everyone talks about getting the stem cells to do this and to do that. And we kind of lose track sometimes on what these cells are. I'm not going to go into them in too much detail today, but I'm going to say a little bit about them. So a stem cell is defined as a self-renewing, undifferentiated, multipotent cell. So self-renewal means that when the cell divides, at least one of those cells will continue to be a stem cell. And the reason that's important is that if you didn't have self-renewal, you'd very rapidly run out of stem cells. So that is just a basic definition of a stem cell, is a self-renewing, undifferentiated cell, which most stem cells have what we call multiple potentials, or are multipotent, which means they have the capacity to form a range of different tissues. And what we do know is that the mesenchymal stem cells, which are derived from, classically, from bone marrow, from the stroma of bone marrow, because of course you also get hemopoietic stem cells in bone marrow. But also you find them in many other connective tissues. You can find them in periodontal ligament, in pulp cells, in the gingiva, fatty tissue, all sorts of places people have isolated stem cells from. But most likely, most of the bone stem cells, the stem cells that give rise to bone, are derived from the bone marrow. And like I say, the key thing that's sort of interesting in the sort of cell biology of these things is actually firstly, they're rather uncommon cells, and secondly, they're remarkably inactive most of the time. They sit in the bone marrow. They're probably in the bone marrow, about 1 in every 3,000 fibroblast-like cells is a stem cell. And it sits there and it doesn't do anything, and it's not dividing, and it just sits there happily sitting, waiting to be called upon. And in sites, particularly in wound healing, then it's called upon, and it's sort of designed to rack up the ante, as it were, very quickly. So you get rapid cell division, rapid migration, etc. So This is actually, you'll probably recognize this as a tooth. It's a mouse tooth, actually, with periodontal ligament and bone. Most of my pictures of bone have got periodontal ligament in them because I'm a periodontist, but that isn't there for this particular reason today. So this is the bone marrow, and within this there are hemopoietic cells, the cells that give rise to all the blood cells, and we know that the bone marrow is a very rich source of, or the main source of hemopoietic stem cells. But also within that, there is a sort of fibroblast layer in which these rounded cells sit on, which you don't really see very clearly in the picture, but these are including the mesenchymal stem cells. And we know, for example, in this tissue, in the periodontal ligament, the osteoblasts that are present die off and are constantly replaced by cells that are recruited from the bone marrow here. So if you label cells, these cells have been labeled with a cell that labels dividing cells. And you can see most of the dividing cells in this tissue. This is the tooth, this is the periodontal ligament, this is the bone, and this is a channel that runs from the bone marrow into the ligament. It's a vascular channel. And these red cells are the ones that are dividing. If you label them and leave it, you can see what happens to these cells with time. And actually, very quickly, within about 6 to 8 days, they've come down the channel now, and they're now migrating. And you can see it's almost like a river flow, that they've come down the channel, and they've now got getting into the periodontal ligament, where they'll subsequently disperse all along there. So that happens in 10 days in a mouse, probably 3 times as long in a human. And that happens all the time. But during wound healing, and I didn't put the data in because I thought it was getting a bit too periodontal if I put the wound healing of periodontal regeneration in, you get a big wave of these cells migrating rapidly to the wound's edge, and that's exactly what happens during bone formation as well. So if you start off with a stem cell, then it has the capacity to give rise to muscle tissue, fatty tissue, fibroblasts, chondrocytes, and osteoblasts. And actually, this isn't just true of bone marrow's mesenchymal stem cells. If you take a cell from periodontal ligament, maybe to your surprise, it's quite easy to get it to make cartilage, which is not something that it ever does normally. If you take it out of its environment, give it the right kick, you can make it make cartilage. But essentially, this stem cell undergoes a stage called commitment, where it starts to go down a pathway, and once it's going down that pathway, to all intents and purposes, it's a one-way process. And if anyone thinks that's wrong, then yes, I know it's not quite right with recent sort of findings in stem cells, but for the sake of this, it's best to think of this as a one-way process. So these cells, as they go through a series of stages, are dividing more and more, and so you get a large number of osteoblasts for each committed stem cell. And you can take these cells and grow them in a dish. They look pretty unpromising, unexciting in a dish, but actually, even within a dish, you can make them form bone. This is actually a piece of bone that's been grown from stem cells, and it has all the bone matrix proteins and indeed will mineralize. It can form cartilage here, which I hope you agree is a nice looking bit of cartilage, actually grown on a bit of dentine, which I think is really kind of interesting for what Frank was talking about earlier. Adipocytes, so these are fatty cells, and then fibroblasts, etc. So what is actually going on during differentiation? And what I'd like to do is take a reductionist view, perhaps a molecular biologist view, which is not my main profession. But essentially, a cell is a single thing. It's really just a protein factory. Everything a cell does is because it makes proteins. So it makes proteins that make enzymes that drive this and that and the other. Every process that a cell does ultimately comes back to the fact that it makes different types of proteins. Obviously, some of these proteins are structural, like collagen. Some of them are enzymes. Some of them regulate DNA. But everything they do comes back to proteins. So a cell is a protein factory, And of course, the protein factory are made from the DNA code. So each protein is coded by a specific gene. And understanding that process is a key to understanding the sort of deep biology of differentiation. So if we took all of the genes from any cell, then you could take classify a number of different genes. So some genes are needed in every cell for the cell to survive. So cells that are involved in cell division, respiration, etc., then all cells would express a certain number of genes, but there'll be a number of other genes that are only seen or expressed or switched on in specific cell types. So a gene may be permanently switched on, so some cells will have genes that are always switched on. And then as a cell becomes more specialized, what it does is actually shuts down permanently most of the genes it's not going to use. So if you're a red blood cell, you don't need to make collagen, because that's not what you do, and so you shut that gene down. Red blood cells are a bad example, by the way, but don't mind. Or you can have genes that can be switched on when you need them. And that's really why I'm showing this little picture. So if we take an embryonic stem cell, a stem cell that can give rise to all the tissues in the body, then you will have these genes that are required for the cell to survive and continue to survive. And then you'll also have tissue-specific cells, and at this case, any of these can be switched on, but they're not switched on at the moment. So any gene that a cell may need in a stem cell, embryonic stem cell is available. Now, once you start to get more specialized, then the sort of next level up would be an adult stem cell, like a mesenchymal stem cell is a stem cell that has the capacity to become a certain number of tissues, but not others. It won't give rise to, say, blood cells. And so some of the genes in that cell are now switched off. And then you come to differentiated cells where, for example, if we take a keratinocyte, there will be specific genes that are potentially switched on, like in the case of a keratinocyte, keratins. Okay, so you can switch a keratin on. Some of the other genes, like collagen, will not be available to that cell anymore. If you take a fibroblast, there'll be a different set of genes, such as collagen. And obviously, if you take an osteoblast, which is where this is going, you'll find that it has a different set of genes which are characteristic of that cell. And ultimately, the osteoblast is defined by what characteristic genes it expresses. So the genes that are characteristic of an osteoblast are essentially ones that are specific to osteoblasts, and a lot of these are matrix proteins that are found in bone. So type 1 collagen, obviously, osteocalcin, which is the most bone-specific of all proteins. So if you've got a cell that expresses osteocalcin, you can say this is an osteoblast. Osteopontin, which is a very interesting protein about which I'm going to say a little bit more, because particularly it's important in acting as a bridge between the matrix and the cell. And so when a cell sticks to collagen, for example, it doesn't stick to collagen, it sticks to osteopontin, which is forming the bridge, hence the name osteopontin. And there are various other proteins that you could say are characteristic of an osteoblast. So a mature osteoblast can be defined as by the genes that it expresses. For example, you could define an osteoblast because it expresses osteocalcin, osteopontin, and other genes such as alkaline phosphatase. And if you want to study this in a laboratory, it's a common way of showing, rather than showing that you've got bone formation, is to show that this cell that didn't used to express osteocalcin is now expressing osteocalcin, and therefore we've turned on the bone-making process. So the issue of how you turn on the bone-making process is, at one level, fundamental to understanding all of the biology of bone formation, and ultimately resulting in lots of developments, essentially, in the clinical management of tissue regeneration, such as bone regeneration. And the factors that stimulate that include soluble signaling molecules. Typically, we would think of growth factors, probably also should say hormones, but growth factors such as BMPs, WNTs, TGF-beta, PDGF. And again, we throw these terms around, and mixtures of growth factors such as you can derive from platelets, for example. But in addition to that, cells also get signals that regulate them, for example, by mechanical loading. So if you stretch a cell back and forth, it changes the genes that it expresses. And we have a good example of that coming up. The cells also talk to each other directly, and then they also get signals from the matrix in which they interact with. So you put a cell on a matrix, which can be anything, it could be titanium, it could be collagen, whatever, then it will be getting signals from that, which may regulate its behavior by changing the gene expression. And you won't be surprised to hear that I'm going to talk a bit more about that. So, if that's the basics of the biology of bone formation, how do we get bone to grow clinically? It's an obvious question. Well, if you asked an orthopedic surgeon, they would say, well, it's not usually much of a problem. If you break your leg, then there are essentially two things, that's the histology of a bony callus, essentially two things you have to do to get bone to regrow. And in this, firstly, you need to oppose the two ends of the wound, and then you have to immobilize them. And the reason for that is that when you place the two ends of the wound together, then firstly, cells that are present on both sides are able to migrate into that space. And secondly, the signals that are required to induce them to make bone are also present and stimulated in a wounded situation. And most of the time, this works very well. And if you don't immobilize it, then the stretching of the cells means that it actually inhibits the bone formation, and instead you get what's called fibrous union, where the two ends of the bone don't unite, and therefore they remain mobile. So it's a bit of a breeze making bone. The problem is that's not where we're dealing with. So we've seen lots of nice clinical pictures. As I've done a few diagrams because I think it makes it reasonably clear. So if we've got a situation like that, where clearly we've seen some very nice clinical photos of this sort of thing, where this is an implant within an alveolar bone crest, then we've got none of the things that you normally need to make bone. So there's no apposition of wound ends. In fact, so if you want to grow bone here, not only is there not bone above it to grow into it, you've got to deal with a completely different tissue. There's an interface between the bone and the connective tissue above it. And actually, as we've seen, immobilization of that wound is also extremely difficult. And all of the surgical things that we've talked about, a lot of those are about really getting a stable, immobilized wound. But the basics are not promising, because the normal things that an orthopedic surgeon would tell you you need to make bone, we don't have. And so we have to go through all sorts of different procedures in order to make that happen. And so this is a kind of terrible, simplistic paraphrase of pretty much everything that's gone in the other lectures. But here you can see a space. We've, in lots of different ways, created a space, and perhaps we've put a membrane there. We've reflected our tissue away from the original place, and we are hoping that the cells from this will migrate into this area and make bone. And I mentioned this interface here because this actually turns out to be a particular problem. In some studies we were doing, which were particularly about periodontal regeneration, but they're just as relevant here, we were asking the question not why does bone form, but why doesn't it form, which I think is kind of a different way of looking at it. And what we were interested in is why doesn't the bone form here? What is it about that? And perhaps it's not surprising, but the superficial connective tissue produces proteins that stop bone forming. And for example, this is an in situ hybridization of a gene called gremlin, nice name, which inhibits BMP, which is a bone-forming growth factor. This is the normal histology, which is horribly out of focus. But this is the in situ hybridization. So here's the alveolar bone. All of this red is where the gremlin is, and this is the epithelium. So this connective tissue is stopping bone growing actively. And actually, when you think about that, it makes sense. Can you think of anything worse than if bone just kept growing into your skin or into your mucosa? It doesn't happen, does it? It just doesn't happen. Well, actually, it does occasionally. Terrible disease called fibrodysplasia ossificans progressiva, which is basically a defect of BMP inhibition. And you get overexpression of the signaling pathway within the dermal layers, and the condition is characterized by these sheets of bone that form basically subdermally, just subdermally. And the terrible thing about it for an orthopedic surgeon is that if you've got a sheet of this bone, then the thing you would want to do is remove it. But the thing that induces the sheet formation is trauma. And so these people get progressively basically encased in bone. It's a terrible condition. And now that there's understanding about the pathways, there's some hope that it can be managed better. So it makes sense that the gingiva is actually specifically, not just inactively, but it's very actively inhibiting bone formation, which creates additional problems. So if we go back to that, then just in that situation, if we just make the space, we may get rather limited cell migration into this area, particularly stem cells. We may not get the right, signals in there. It's competing with the inhibitory signals, and clearly, on its own, that straight GBR type approach is actually very ineffective, not least because of the difficulties of immobilization. So we will take all sorts of different ways, and there are lots of different strategies to try and make this happen, to make stem cells in there, to make them differentiate, to give them the right signals, and to immobilize the wound to allow it all to happen and to heal. And I think we've pretty much seen most of the different things that you could put in there today. The only thing we haven't really talked about that you could put in there would be a growth factor. Well, we talked about PRF, but you could have put in a recombinant growth factor like PDGF or BMP, firstly, if it was licensed, and secondly, if you could afford it. But that's about the only thing we haven't really talked about that you could put in there. And it's quite interesting when you look at the different ones, they all fit into that model, and for different situations, there are different good ways of achieving that. I've made it look very simple, but obviously it's not. And quite often, an easier way of making that happen is actually to do it before you place the implant, of course, because at least then you don't have the problem of the interface between the implant and the bone. So that would be an easier situation to manage. Now, Simon showed some lovely cases where bigger and bigger, and the more you think about that, the more a challenge these things become. So we are going to need a range of different things, and often biomaterials. The great thing about biomaterials is they tend not to have the morbidity associated with them than grafts do. But obviously, they're not appropriate for every situation. But a biomaterial will be used because we can promote cell migration, Firstly, by making the space, but particularly by using a material that will allow, we talk about scaffolds, but essentially you need cells to be able to migrate on surfaces into the wound, to provide the right signals. Now, the right signals turn out normally to come from the cells that migrate in. The body has an amazing capacity to provide its own signals. And actually, most of what we do in any of this sort of regenerative stuff is to facilitate the body's own healing mechanisms. Excluding inhibitory signals essentially means you've got to make enough space to keep the superficial tissues out the way and to improve immobilization. And when we look at what biomaterials do, then there are probably a range of different things. At its simplest, the biomaterial is biocompatible, so it forms a good barrier. And the best example that I could come up with was PTFE, and the PTFE with titanium to reinforce for, this, for shape is a very kind of effective way often. But also materials can do a number of other things, supporting cell attachment. And if you think about osseointegration, then the tissue doesn't just grow up to the titanium, it does actually attach to it. And that, I think, is quite an important thing that it does. But obviously, a lot of other biomaterials that we use support cell attachment, such as collagen, such as bio-os and other alloplastic materials. And a number of materials support cell migration. I don't know whether they promote cell migration or just support cell migration. Maybe for me that's a bigger deal than it is clinically. What we'd probably like, because it sounds good, and actually when you've got three different ways of doing these things, you tend to throw all three at them. But the evidence that actually you get an increased effect is not that strong. And the evidence that anything is directly inducing the bone cells isn't very strong. And it was quite interesting listening to Frank's talk, where really his bone block didn't appear to perform any better than his dentine block. His dentine block, dentine will contain some growth factors, but essentially it's an avascular mineralized material. It's not very biologically active. Put a bone block in there, you assume you're getting all the benefits of the osteoblasts that are in there, et cetera, et cetera. It may not be the case at all. So most of what we're doing with the material is to facilitate the body's own healing process. So with that as sort of background, we've been interested in all sorts of things that are related to how you might develop things clinically from understanding the biology. And by happy chance, a few years ago I hooked up with Paul Buxton, who's head of the Translational Cell Biology Research Lab at Geistlich, and we've done a number of things together, and they're funding another very interesting project of ours at the present time. And actually this work that I'm going to talk to you about now is some fairly simple work, which was originally started at Geistlich's labs, but it was so sort of striking that I think they felt it needed independent verification. So we've actually done most of the data I show you is actually our data. And you know, one can understand why they might want us to do it. So what are biologically or clinically relevant parameters which we can measure in the lab? So attachment, proliferation of cells on membranes, migration of cells on the surface of membranes, and then the effect of the collagen membranes on the stem cells and how they behave on the stem cells. All of those things, for us, they're pretty easy experiments to do, actually, because essentially what we're doing is taking a membrane, we're growing the cells, and then we're measuring an outcome on them. So essentially, we know there are many collagen membranes around for these applications. And although the details of how these things are made are not clear at all, and I should say personally I have no idea how they make the Bio-Gide membranes, even though I've done a reasonable amount of work with Geistlich. The first time I ever visited Volhusen, where Geistlich are based, or their research labs are based, etc., I was so struck. I was shown all of these different membranes. There's just like a desk load of them, which all things that they'd made in their labs. And I was just so struck by how different they are, both particularly and obviously in physical properties. They're amazingly different. You know, you wouldn't have even guessed they were necessarily the same material. And because you can make collagens in so many different ways and adapt them, it sort of begs the question, how does that affect cell interactions with them? So these are a list of some of the, I'm not sure whether there are any here. Anyone got a favorite that isn't on this list? There's quite a long list, and these are the different membranes we've tested, essentially. And when you look at this list, sorry, that's a repeat of that, which is not too exciting. They're essentially, you can class these into two types in their basic manufactures. Some are taken from a tissue and processed. As I say, personally, I have no idea of the processing that these things undergo. So if you look at these ones are all derived from the tissue. I believe it's very easy to find out what the Geistlich one is because it's in the patent, but you know, you've got to go and look through the patents. It's not published. Or in the other ones, they are collagen that's been extracted from a tissue and then been reassembled in a different way. And I think conceptually that's quite a different sort of thing, because here we've got, say, pericardium is quite a common one, where you've got a sheet which will tend to be organized in a particular way. And the other ones tend not to be. And you can see this quite clearly if you take some cells and you seed them onto a membrane and grow them for a bit, then the membranes that are not reassociated but extracted directly from processed from a tissue encourage directional growth, whereas these are cells that have been stained fluorescence, just to give you a nice green color. But the cells the membranes that have been reassociated have a completely different pattern of cell migration. And when you look at them, you can look at the different membranes and you can see the Bio-Gide one's very directional. Some of the others are quite different from that, but there's a range of different responses. But essentially, the ones that are derived from tissues are quite directional. And if you look at the ones grown on Bio-Gide, you can see this is actin staining, so it's the cytoskeleton of the cells, so you can see how well they're organized up and down, basically orientating along and migrating along these collagen sheets. So in this simple assay, you can distinguish essentially the two groups of different membranes. Those that are retaining their native orientated structure and those that don't, such as this one here. So then we started to look at the differentiation of mesenchymal stem cells into osteoblasts. And like I say, if you grow mesenchymal stem cells in culture, they will tend to grow, and actually you then have to do different things to them to make them into osteoblasts or cartilage cells or whatever, or fat cells are the three easy ones to do. So when you do this, what we're going to do is we're going to put the cells onto the membrane, and we're going to look at the expression of these genes, because these are the characteristic signals that say, I am now becoming a bone cell. And actually, the data I'm going to show you is really mainly with osteopontin, a bit on osteocalcin. And we have done this, because of the nature of the data, we've done it on 5 different cell lines, because each cell line is a primary cell line. It's extracted from a human. You can buy these cell lines. They're very expensive, but less expensive than having to persuade someone to have an iliac crest biopsy for then growing their cells. So we buy these cell lines, and between us, we've used 5 different cell lines derived from different donors on all of the different collagen membranes, and then we looked at the expression of genes, which is analyzed by PCR. So for me, the thing that says bone cell or not is osteocalcin expression. And this is sort of quite striking. So this is basically normalized. It's fold different. So this is control, and you can see this one here is 5 times above control. That sounds like a lot. It's not, for gene expression, it actually isn't that big a change. So in our normal cultures, without kicking them with anything else, then just growing them on this membrane here increases their osteocalcin 5-fold, which is indicative of osteoblast differentiation. And you can see now why they wanted corroboration, because you would never predict that only one of these membranes actually has this effect. You really wouldn't predict it. And if you do it over a time course, then as expected, as you grow them with time, day 7, day 14, these are different controls on this one, then really at a late stage of differentiation, you get this big increase in osteocalcin. Now, when you look at osteopontin, it's even more striking, and the data we have are more comprehensive. Again, if you look at the time course, you see that osteopontin comes on particularly at day 14. And if you look here now, the numbers are much higher. So in this one, this is nearly 25-fold higher than controls, and 2 different days. But the late day, you can see this huge increase in osteopontin compared with really all of the others. And another data, another different cell line this time. Again, you can pick out the Bio-Gide straight away. Occasionally, actually doing this experiment is a bit of a fiddle because the membrane is not a very easy place to get the RNA off. So occasionally you get some data where nothing happens. But essentially, you can see the same pattern. And another one, in this cell line, you get this huge fold increase compared with all the others. Now, that looks dramatically different to all the others, but the reason it is you have to remember this is fold difference. And if you start off with a very low level of expression, then the actual change is huge, but the relative amount is probably not very different to the expression that you see in the other cell lines. Now, you probably think I've come here to diss all the opposition. I honestly don't know what this means. The interesting thing is, of the genes we've looked at, the one that's striking is osteopontin. The osteocalcin works, you're clearly getting differentiation, and osteopontin is indicative of differentiation, but it also does something different, which I mentioned, which is cell attachment. And in this particular case, what you're looking for is cells to attach and migrate on these membranes. Hopefully in a directed way, because you potentially could exploit that further and think, well, if I can direct the cell migration, then that could be additional benefit. And osteopontin is the protein that does that. And for some reason, which we don't know, this membrane seems to really promote cell attachment and migration in a way that the other membranes didn't. So collagen membranes differ widely in their derivation, manufacture, physical, biological properties. And for me, that was the biggest surprise, because kind of collagen's been collagen, been collagen. In some of the other experiments we're doing, actually sponsored by Geistlich, we're using collagen gels, but it kind of was just thinking maybe we should use different collagen gels, but anyway. So one membrane type tested, which should remain nameless, performed completely differently to really any of the other types when we were looking at their effects on mesenchymal stem cells, particularly in relation to osteopontin expression. And I'll be absolutely honest, I have no idea clinically what that means. It's a real challenge to say, is that membrane better than the others? Does it work better? Doing a clinical study to do that is actually quite difficult. It would be very hard to show true differences between different membranes unless you had a model that was particularly demanding. A clinical model that maybe your very large defect, give me the largest defect you can, Simon, and maybe that's the sort of experiment you'd have to do. And of course, And it's interesting because we all do it, and Frank was doing it earlier, I noticed, that when you put up a study and it says, you know, if you use a membrane, you get a better result. And actually, maybe you don't know what differences the different membranes make. I don't know. I'm not trying to sell you Bio-Gide, and it may be that actually this property isn't a desirable one, but it does look to be a very desirable property. But I think it's important to say that I don't know how important clinically these findings are, but it's certainly interesting. It gives us a sort of window on the opportunity to try and work out what it is about a collagen membrane that actually has these properties, because we've tested some of the odd-numbered ones in the data, which you may have noticed are actually test collagens that they have at Geistlich. So I'd like to thank you for listening. Thank you for your patience, because I know this is a bit of a diversion from the main event. Particularly, I'd acknowledge Neelam Gurav, who is a postdoc who did really all the work in my lab, and then the people at Geistlich, Paul, Marco, and Christina. And of course, massive conflict of interest. The study was funded by Geistlich. So thank you all for listening, and happy to answer any questions. Thank you.